Can't stay in one spot. Albie asked me to give a sort of a brief introduction to Alexander disease and then talk about some work that we've been doing trying to characterize, thinking about characterizing Alexander disease as, among other things, as a central nervous system inflammatory disease. And I'll show you the data from that in a few minutes. So, as many of you know, W. Stuart Alexander, whose name was given to this disease, not by Dr. Alexander, actually, but by other people, in 1949 published the first report of a child with Alexander disease from the London hospital. And after 1949, there were a number of clinical and pathology papers in the medical literature on Alexander disease. But it really wasn't until 2001 that a group of us, spearheaded by Mike Brenner, found that mutations in glial fibrillary acidic protein are associated with and actually causal Alexander disease. This, as many of you know, is a filamentous protein that's found in astrocytes in the central nervous system and all over the central nervous system, in the brain, the brainstem, and the cerebellum, and in the spinal cord, so everywhere. This is actually an older slide. There are more up-to-date ones, but it shows you the protein itself, starting at one end here and going through in the other end over here, and showing you the number of mutations. Each of these numbers here represent a single amino acid change in the protein structure. And as you can see, there are many, many different, each patient has a single one of these, but there are many patients, and these mutations actually run all over the molecule. Nevertheless, most of these people present with Alexander disease. So it's a mutation in the GFAP gene. It's a dominant mutation. That is, people with Alexander disease carry one normal and one mutant copy of the gene. Most of these mutations are new mutations. That is, if you look at parents of many of the Alexander patients, you'll find that they're normal. You won't find any mutations in the GFAP gene. So it means that the mutation arose spontaneously, either very early in development or as a germline mutation. However, there are a few families in which the mutant gene can be passed on to the next generation. And the way that's happened is that, as many of you know, there are some people who develop the clinical signs and symptoms of Alexander disease later in life, past their childbearing years. And they're not known to have Alexander disease, and so they have children. And since it's a dominant mutation, there's roughly a 50% chance of passing on the mutant gene to the children. Okay. As in many genetic diseases, people with Alexander disease represent a spectrum from newborns all the way to people who are much older. And as is the case with many genetic diseases, babies who get this disease progress much more rapidly and have a much more severe clinical course than people who present later in life, as teenagers or as young adults, for example. Adult patients usually develop clinical signs and symptoms a little differently from the babies. And many of them develop problems with brainstem functions, that is, swallowing or speaking. Some of them have problems with balance because of cerebellar abnormalities. All right, first, a few words about astrocytes, because we know that the GFAP gene is expressed only in astrocytes in the central nervous system. So this is a disease caused by a mutation in one cell type. It's not expressed in neurons, and it's not expressed in oligodendrocytes, the myelinating cells of the central nervous system, or any of the immune cells. Astrocytes come in a lot of different flavors and shapes and sizes in the brain. This is from a rat brain, but it's the same idea in human brain. They take many, many different forms. They have now what's known to be a huge number of critical functions in the central nervous system. I've listed them here, and I'm not going to go over them, but you can look and read some of these. This is a human astrocyte here, and you can see this is stained with an antibody we use for astrocytes, and you can see that it's basically a round cell. Here's another one over here with many, many, many fine processes that's attached to blood vessels. Here's a blood vessel, here's a blood vessel, and here's a blood vessel. Astrocytes basically cover all of the blood vessels in our brains and play a critical role in managing and regulating cerebral blood flow. So when something goes wrong with an astrocyte, there are tremendously critical consequences for all of the other cells in the central nervous system. And many of these normal functions that we associate for astrocytes are in fact disrupted in Alexander disease. And we can use that to explain some of the problems that our patients with Alexander disease develop. Here's an MRI that many of you are familiar with, an axial flare MRI, showing these white matter changes in this particular person in the frontal lobe. And you can see it's largely found in white matter. Sometimes people present with occipital lobe symptomatology and changes, sometimes frontal lobe changes. If you look at the brain under the microscope, you see, particularly in young patients, a tremendous loss of myelin. And myelin, as you know, is the fatty material that sheaths axons and allows axons to conduct electrical impulses. Without this material, axons cannot conduct, or conduct very, very slowly. This is from a child with Alexander disease, and you see a loss of myelin. Myelin should be blue in this picture, and there isn't any at all. And you see all of these red accumulations. These we call Rosenthal fibers, and they're accumulations of this mutant GFAP plus other proteins that occur inside astrocytes. So this is a brief description of these Rosenthal fibers. This is what they look like. They're little red things like this. This is an electron microscope picture. So we look at this under extremely high magnification. What you see here in this astrocyte, see these linear things here? These are GFAP filaments, and that's what they normally should look like. And you'll find these in astrocytes. However, there are these blobs. We call the pathological term blobs. So, that sit on top of these, and these are the so-called Rosenthal fibers. And you see these typically in astrocytes that have accumulated a lot of GFAP. Okay, so the accumulation of wild-type, normal, and mutated GFAP in astrocytes causes these astrocytes to assume very strange shapes and alter their functions so that they're no longer able to regulate all the important things in the brain that they normally do regulate. And here's an example of a normal astrocyte I showed you earlier. These are 3 astrocytes. This is actually from one of our mouse models, one of Albie's mouse models. These astrocytes look like this. If you compare them to something like this, you see they're tremendously changed. We got these because we can make slices of the mouse brain and put small electrodes into individual cells and record from them and find how different they are from normal astrocytes. And then we squirt a little bit of green fluorescent stuff into them so that you can actually see what these guys look like. And we've seen some of this in human brain also, that these astrocytes change shape tremendously. They lose a lot of these fine processes and assume these very odd shapes. They also lose their ability to manage the extracellular fluid in brain, particularly some of the neurotransmitters and particularly the ion potassium, that needs to be regulated in a very, very narrow window. Okay, so we saw this. The astrocyte dysfunctions lead to demyelination, or in babies, a lack of normal myelination. They also lead to neuronal damage, not necessarily neuronal death, but neuronal damage. And at least in the mouse brains, we can find that many of these neurons are now becoming the type of neurons that one sees in epilepsy. And we know that many of our Alexander patients develop seizures. So the neuronal dysfunction caused by these changes in astrocytes leads to things like seizures, changes in motor function, behavior, and cognition. And so that's sort of a general introduction, and you'll see some of these themes repeated over and over again by most of our other speakers. I'd like to talk about something that's new, and that is inflammation in the Alexander brain. It's never been thought of really as an inflammatory disease. But there are some elements of inflammation in the Alexander brain. And this was first suggested by Tracy Hageman and Albie in a paper several years ago now, in which they looked at the genes that are expressed in these mouse brains. And they found that a number of the genes were genes of inflammatory molecules. A couple of years ago in our lab, we did a study on another one of the mouse models, and I'll show you some of the data. There was a marked inflammatory environment. So what do we mean by inflammation? And that can mean a lot of different things to a lot of different people. Many of you think of inflammation as, an infection, for example, or an autoimmune disease like multiple sclerosis, or other things like that. Well, we have certain specific definitions of inflammation, and I'll show you what we mean by it in Alexander disease. This is not supposed to have you read these things, because you can't if your eyes are as bad as mine. But one of the definitions of inflammation is that there are high levels of molecules that are associated with immune function. We call some of these things cytokines or chemokines, and they're made usually by inflammatory cells. In this particular case, in mouse brain, and we think also in human brain, many of these are made by the astrocytes themselves. And this is just to show you, these are actual protein levels of these various inflammatory molecules. We've looked at a few human brains, and these are frozen tissue brains, and compared them to some brains of normal people of the same age who don't have any neurological problems. And if you just focus on the gray here versus the blue and the red, you find that there are many of these inflammatory molecules whose levels in the Alexander brain are much, much higher than one finds in the normal brain. We've looked at these more extensively in mouse brain and find that some of these are actually expressed by the astrocytes themselves. And some of them are expressed by, as I'll show you, the intrinsic inflammatory cell in the brain. These are called microglia. I'll come back to that in just a second. So that's the first thing, that both the mouse and our preliminary studies on human brain show us that these molecules are highly elevated in the Alexander brain. The second thing we think of when we think of inflammation is the entrance of immune cells into the brain, particularly things like lymphocytes or neutrophils or macrophages from the blood into the brain. And we've looked at both mouse and human brain for a group of cells called T lymphocytes and found them. Here, these are immunostains, so they're dark brown are the lymphocytes. The blue here is just a counter stain. So here's a section of human brain, also over here. And these brown cells are the T lymphocytes. It's not a big T lymphocytic inflammation in Alexander brains, not nearly as much as you would see in some autoimmune brains, like multiple sclerosis. But they are there, nevertheless. And the third thing that we mean by inflammation is an activation of immune cells that are intrinsic to the brain. Not only does the brain have neurons and astrocytes and oligodendrocytes, are all related to each other, but they also have these cells called microglia. And those cells are professional immune cells. They enter the brain extremely early in human and mammalian, generally, brain development, and then stay there for life. We all have zillions of these guys in our brains, and they are constantly patrolling their own little domains for foreign objects. And they respond to viruses and bacteria and foreign materials and things like that. And they help us ward off infections, for example. What you find in a normal brain, and this is a normal human brain, the microglia are in green here. The red, by the way, is GFAP. And the blue are actually cell nuclei. So these cells are very delicate, small cells in a resting brain. They're not doing a lot except sort of patrolling their territories. In the Alexander brain, however, you see a tremendous activation of these. These guys are extremely big. All of these guys are microglia. They become extremely large. Some of them turn into macrophages, which eat up dead tissue in the brain. And these are also starting to express some of these inflammatory molecules. And you see this in many areas of both the mouse and the human Alexander brain. So we have, I think, very strong evidence for an inflammatory response to the abnormal astrocytes in the brain. So what are the consequences of inflammation in the Alexander brain? One is that we know that inflammatory molecules can damage myelin and myelin-forming cells, the oligodendrocytes, and that can lead to demyelination, or in the case of babies, an abnormal or an inhibition of the normal myelination that normally takes place during the first few years of a baby's life. And that doesn't happen in infantile Alexander patients. The second thing is that inflammatory molecules can damage neurons. There's a very, very slim medical literature on this, however. And I don't quite understand why, but there is a very small literature on the effects of some of these molecules on neurons. Some of it's been done in culture. Some of it's been done in slices of mouse brain or rat brain. Basically, you can alter neuronal structure and alter particularly neuronal function. You can alter synapse formation. You can alter the physiological properties of the neurons and make them more excitable. In that way, turn them into cells that may contribute to seizures. And finally, just a question for our experts, but also for everyone to think about, would decreasing the inflammation in the Alexander brain have any therapeutic effects? That's something I think that's worth thinking about seriously. You're going to hear about clinical trials today, and maybe we could talk about this then. I don't think that this inflammation is going to change levels of the mutated GFAP, and you're going to hear about some ways that we might be able to do that. But given that patients are diagnosed at some point, possibly well into the stages of their disease, they presumably have already started to build up a big inflammatory response in the brain that causes these other things. And my question, I guess, that I'll leave everyone with is it worth trying to think of anti-inflammatory therapies as an adjunctive type of therapy for some of our Alexander patients? And many people over the years in our lab have contributed to this, as well as a set of wonderful collaborators over the years. We've worked with very closely at UAB and Harvard and Wisconsin and Einstein and Rochester. And I've been very fortunate in having them, with us and working closely with them for many years. Thank you very much.
Okay, so we have time for some questions.
Yes?
So in terms of this question of whether it would be protective to tone down inflammation in the patient brain, what is known about, presumably this pathway has been manipulated in other neuroinflammatory disorders, and I think it's hard because early on maybe it's protective, but later on it could be damaging. So how would we start to think about resolving the timing issue and when to?
Well, I think that the timing issue is really critical. And I think that by the time most patients with Alexander disease are diagnosed, it's somewhat far along in the clinical course. For a number of reasons I think that we all understand. So I think, by the time people are diagnosed, these are presumably having, more destructive types of problems than protective sorts of problems. I think, we're just starting out on this, and I'd like to look at a lot more frozen tissue to try to characterize exactly what's there. I mean, we know a tremendous amount about the inflammatory response in some of these mouse models, but as we all know, mice are different from humans. And the immune systems of mice are quite different from human immune systems. And so we've already seen, in looking at just a few Alexander brains, some differences between the types of molecules that you see in human brain and the types of molecules you've seen in mouse brain. I think, obviously, once we get a better handle on exactly which sets of molecules are upregulated, then it would be obviously, one next step would be to look in CSF and see if there's any sort of correlation there. Yes?
Just sort of like limited treatments, infusion treatments for the inflammation. And within just a few weeks, my daughter's neurologist was shocked that it actually caused things to progress rapidly.
What sort of treatments were those?
I don't have the name, unfortunately, but we did, it was like a 3-4 days, she was in the hospital, and I'll have to get the name, but yeah, within just a couple weeks, we
Yeah.
were hoping to see something, like slow down or whatever, but she actually had more damage, the spots were big.
Yeah, I think that it depends critically on the type of treatment. I can imagine that some types of treatments would make it worse, and other treatments, we don't know, could potentially make it better. So what we're trying to do now is to sort of figure out what the targets are. And I think that, giving, I mean, there are a lot of general anti-inflammatory treatments that we give our patients with autoimmune diseases, for example, steroids and worse drugs like methotrexate and a bunch of other things. But I think that it's not clear to me as yet which ones are going to have appropriately positive effects. I just don't know. So I can imagine situations in which you can make people worse with certain types of anti-inflammatory treatments. So thank you for your comments. I can certainly imagine that.
My question was actually related to this, like what kinds of treatments you were thinking of, but you can answer that.
I think there, in general, in treating patients, say, with inflammatory diseases, there are two sort of general types of treatments. One is to give a blanket treatment, like corticosteroids. Right? The other is to give a specific treatment, like if you find that, for example, there's a particular interleukin that's highly regulated, there are drugs that can be given that are antibodies to those or can block receptors to those. So it ranges all the way from very highly specific drugs to target certain specific pathways to very generic blanket drugs. And I just don't know yet what's going to be appropriate, and you can't know that until you actually know what's going on and what sort of things.
I'm sorry, is there any research on the horizon for using these different types of treatments, or is that kind of the next step?
Well, I don't know. I think you can ask Amy has a comment about that.
Yeah, so just to add to that, so many patients that we've seen have actually been put on steroids prior to the diagnosis being made, because the physician in the hospital, it takes weeks and months, and you all have better experience in understanding how long it took to get a diagnosis. And we found that a lot of patients were being put on steroids and had some clinical improvement. And in sort of going through this over the last 2.5 years, so the symptom that seems to have responded the best to steroids is mostly symptoms of the vomiting, not necessarily motor, not seizures, not those types of things. But so some families and some patients that have had, especially brainstem lesions that are enhancing, steroids or the corticosteroids are something that we use in multiple sclerosis all the time, and it decreases enhancement or contrast into the brain. And so we've seen some improvement in patients who mostly vomiting, the steroids have improved. Everyone feels a little more energetic on steroids, so steroids have some added benefit in that way, but we haven't seen gross motor improvements or necessarily speech or other bulbar symptoms on steroids. So I'm not advocating steroids as a treatment right now for Alexander disease, just to share our experience, and some people don't know that I've suggested steroids in certain situations, and sometimes we'll do a trial and say, okay, I know that you're targeting this particular treatment for yourself or your child, let's see if steroids with very clear starting and stopping points, saying, okay, if it helps that symptom, then we're going to continue. If it's not helping that symptom, then we're going to stop, because steroids have some side effects. Not clear whether your daughter got steroids or maybe IVIG as an anti-inflammatory. And then there's also been a consideration, Albie, I don't know if you want to mention, that at one point people were considering using an MS drug, and you're referring to
Right.
another type, it's not really an MS drug, but an IL-6 blocker and some other
Right.
therapies that are used in these other diseases, but we haven't done those in patients with Alzheimer's disease yet.
I think if we were to find extremely high levels of certain specific inflammatory drugs, it might be worth thinking about that.
Can I just say real quick?
Sure.
5 years ago, my daughter was on hospice. She was 12 years old. We put her on an anti-inflammatory diet. Her GI doctor had suspected she had celiac disease, but all tests were negative, according to the biopsy. She had asthma, just horrible blood work. Long story short, we followed like a Walsh protocol of gluten-free, dairy-free, low sugar, and all of her blood work is perfect. She no longer has asthma. She's out of her wheelchair. 17 years old. She's going to high school. It's just a humongous improvement just by changing her diet. And we do follow like a multiple sclerosis type of diet. for her,
Right.
But it's helped her
Right.
so much.
Good. I'm glad to hear that.
I got her all medication. She doesn't have seizures anymore.
Okay, so I know, there may be other questions, but we should move on, and but I encourage you to bring up those questions during